Hi students, good morning, all the students of class 10. So once again come back to your study mode after your summer vacation. I hope that during the summer vacation, along with taking some rest, you have done your studies, you have done your holiday homeworks and the assignments that has been given to you. Now, before the summer vacation, I have completed 2 units. Unit 1, the basic biology, and Unit 2, the plant physiology. Up to that portion we have read. Now, if you are having any queries regarding that, we will solve it out in our Google Meet class. And the link I will provide you in the verso. And today, I am starting with the Unit 3, the first chapter, that is the circulatory system. Today, I will give you a brief idea about the circulatory system. And if you are having any queries regarding that also, you can ask me in the Google Meet. Clear? So, let's start with the circulatory system. Now, before starting the circulatory system, I hope that you have a little bit knowledge about the system because already this chapter is there in your previous classes also. Then also I will try to give out a little bit explanation of the previous one, and then I will continue the details of this chapter, that is the circulatory system. As we know that the circulatory system is mainly concerned with the transportation of the substances like the oxygen, respiratory substances, glucose, etc. in our body. It is acting as the circulatory medium. Now, as I am using the word circulatory medium, that means one component is there that acts as the circulatory medium. So, here I told you about the definition of the circulatory system, and the circulatory medium is the blood. Am I right? Blood helps in transporting all the substances throughout your body and it circulates within your body. Now, if I come to the components of the circulatory system, here we are having 3 main components. What are the 3 main components of the circulatory system? Let me write it down. The 3 main components of the circulatory system are, number 1, the body fluids. Under this body fluids, we are having number 1 as the blood. Number 2 is the lymph. And number 3 is the tissue fluid. We all know that this circulatory medium needs a channel through which it will flow. The same is true for this other 2 also. So, the circulatory medium for the blood flows within the blood vessels. Similarly, the lymph flows within the lymph vessels. And the tissue, it bathes the body cells. It bathes the body cells. So, the second component of the circulatory system are the vessels. We can have the blood vessels here. At the same time, we are having the lymph vessels. Or you can say as the lymphatic vessels. Clear? And the third one, not the least, that is the pumping organ of our body, that is the heart. Clear? So today, mostly I will deal with the body fluids, that is the blood, lymph and the tissue fluids. Before going to in the details of the blood, let me have an idea about the relationship between these 3. Relationship between these 3 fluids, that is the blood, lymph and the tissue fluids. Tissue fluids is also known as the interstitial fluid. So, for having a brief conception, I am trying to draw out a figure over here that will help you to have a rough idea about the relationship between these 3, that is the blood, the lymph and the tissue fluids. Here you can see I have simply drawn 3 figures. Okay, as red I have drawn here the one of the blood vessels, the thinnest blood vessel. This is the capillary. Okay, we know the blood flows in the capillaries, and this is the thinnest one that comes close to the tissue cells. This one I have tried to draw here the body cells. I have shown here by this figure as the body cells, and this one is the lymph vessels. Clear? The green one. Right? Clear? Now then, what are the tissue fluids, isn't it? I simply told you the tissue fluids but the body cells. That means when the blood is flowing within this capillaries, then the plasma comes out of the blood to form the tissue fluids. That means it's coming out from here. Okay, this is coming out from here, from the capillaries into the space that is between the body cells and the capillaries. The plasma is coming out and forming the what? Tissue fluids. Now the glucose, mainly the glucose, that is the nutrients, the oxygen, respiratory pigments, this all will diffuse from the capillaries to the body cells and burn it. Clear? Now the same, after it goes to the body cells, they will produce some metabolic waste. That metabolic waste, taken in the form of maybe CO2 or other metabolic waste, will again come back and diffuses again into the capillaries. Again this capillary will take the waste along with the blood and remove it out through the excretory system. Am I clear? Now within this time, some of the tissue fluids also leaks out from the body cells, the plasma, and then they are absorbed by the lymphatic vessels or lymph vessels, and here they are found as lymph. They are found here as lymph. So they form the lymph. So the fluid that flows within the lymphatic vessels, they are known as the lymph. And the fluid that burns the body cells is known as the tissue fluid or interstitial fluid, and the blood that remains confined within the capillaries is known as the blood. So here you can understand the relationship between the three body fluids that are found in our body, that is the blood, the lymph and the tissue fluids. Now as we are dealing with the circulatory system, so our main focus is in the blood, that is the main component of the circulatory system. Now let us come with the blood. So blood, first I am writing this one as you are reading from your junior classes. The components of the blood. The blood is having two main components. What are that? One is plasma, another one is what? Corpuscles. Plasma is the liquid part and corpuscles is the cellular part. Plasma comprises about 55% of the total and it comprises about 45%. Clear? The cellular part, that's remains suspended in the lipid part, that is the plasma. Now this plasma, out of that 55%, 90 to 95% they comprises of water, and the rest 5 to 10% they comprises of many things. What are the many things? Many organic substances. Maybe it can be protein in the form of some protein like globulin. It may be in the form of some nutrients. What are the nutrients? Fatty acids, monosaccharides, disaccharides. It can be amino acids. It can be some traces of hormones. Okay, some ions like sodium, potassium, etc. and so forth. Clear? This is the composition. That is the distribution of the inorganic matters in the plasma and the organic matter in the plasma apart from the water. Now the another part, that is the cellular part, due to which the blood is getting its color also, the red. That is the three. Here we are having three. What are that? That one is RBC, next one is WBC and the third one is platelets. Let's come first with the RBC. Okay, very fine. The first one is our RBC. I hope all of you know that the another name of RBC is erythrocytes. WBC is leukocytes and the platelet is thrombocytes. Now this RBC, we have to read about its formation. If anything has its formation, then there is a breakdown of that one also. So here I am going to give you two important terms regarding their formation and their breaking down. Before that, I'd like to have a brief recap of things that you already know, just about the lifespan of the RBC, the shape of the RBC as you already know. Then also I am trying to give out to you a right idea about that. Let me draw here a smear of blood. Okay, just I am trying to give you the shape of all the components of the blood, the cellular components. Here I am drawing, as it is red in color, so it's better to draw in the red, isn't it? Okay, what are this? Yes, these are the RBC. They are biconcave in shape, and from the, if you have a surface view, from the clear view, then they will appear round more or less, but actually they are biconcave in shape. And if I come to the lifespan, they are nearly about 120 days they live, and you can see that they don't have any nucleus, isn't it? They don't have, being a cell, they don't have any organelles in it also. Now the question is why? Why they don't have any organelles like mitochondria, endoplasmic reticulum, or the most important, that is the brain of the cell, that is the nucleus in it. There is a point why they don't have, because to increase their efficiency for carrying the oxygen. We know that hemoglobin of the blood mainly helps in the carrying of the oxygen. It helps as a oxygen carrier, and more RBC means the body cells will get more and more oxygen, and hence we will get more and more energy after respiration and for governing our work. We can do our work. So they are devoid of any organelles, they are devoid of nucleus, so that they can have a high efficiency for carrying of the oxygen to the each and every source of our body. Am I clear? Okay. After that, if I come to its formation and breakdown, then what is that? There are two important terms. One is erythropoiesis and another one is hemolysis. The first one is the formation and the second one is the breakdown. This one is the breakdown. The formation of the RBC occurs in the liver, in the spleen, in the embryonic stage. But in the adult stage, it mainly occurs in the bone marrow. So the formation of the RBC, whether in the liver, in the spleen, or in the bone marrow, that is called the erythropoiesis. And the breakdown of the RBC in the spleen and the liver, or the one out of the dead, whatever may be, that is called the hemolysis. Now, what is the fate after RBC is broken down? It is broken down in two parts, as we know that RBC is having the hemoglobin part and the globulin part. Then what is the fate of these two parts? What happens to both of them? This hemoglobin is again utilized in the by the bone marrow for the formation of RBC. This is again utilized. Who utilized it? The bone marrow utilized it. The bone marrow again utilized it for the formation of the RBC. Then what happens to the globulin part? They are converted to bilirubin and biliverdin. They are converted to bilirubin and biliverdin and are sent to the liver, where from they are excreted out as bile. So bilirubin and biliverdin, these two components, where from they are excreted out from the liver as bile. This is the actual fate of the RBC. So here see two important terms are there: erythropoiesis, hemolysis, and the fate of the iron part and the protein part, that is the globulin. Clear? This is all about, I just explained about the RBC. Now yet there is another important thing. We know that RBC is having a high affinity with the oxygen, so it binds with oxygen and helps to carry it to your body cells. And this they do so by forming a compound that is known as, when they combine with oxygen, they form a compound which is known as oxyhemoglobin. They form oxyhemoglobin. They form oxyhemoglobin when they combine with oxygen. Hemoglobin, when combined with oxygen, they form oxyhemoglobin. But sometimes if there is more carbon monoxide, then what happens? Then instead of binding with that oxygen, they bind it with the carbon monoxide. And at that time, they form a very stable compound, that is, instead of forming oxyhemoglobin, they form a very stable compound that is called carboxyhemoglobin. They form the carboxyhemoglobin. I am writing it above here only so that you can understand better. Instead of, sometimes they combine with carbon monoxide. If they combine with carbon monoxide, instead of combining with the oxygen, if they form with carbon monoxide, then they will form carboxyhemoglobin, and that is a very stable compound, and that is fatal for us also. Got it? So for that reason, it is better that if it combine with the oxygen and form oxyhemoglobin, then in our body the functioning will be alright. But if by any chance or any circumstances, if the amount of carbon monoxide is greater, then they have an affinity to bind with that only, and in that cases that will be a fatal for us. Is that clear up to this much? Okay. This all I deal with the RBC. I said about their lifespan, I said about their shape, I said about their formation, and I also explained about their breakdown. Clear? And this here I have shown how they look like. At the same time, I also said why they don't have the nucleus and other cell organelles. This is a part of the question which is very frequently asked in the questions, that why RBC are devoid of nucleus and the cell organelles. Okay, so I have explained the answer for you also. Next is coming our great deal, that is the WBC. And we know that WBC are having two. One are granulocytes and another one is agranulocytes. So let me write. One is granulocytes and another one is agranulocytes. Under granulocytes, we are having again three. What are that three? The first one is eosinophil, basophil, and the third one is neutrophil. Eosinophil, basophil, and neutrophil. And the last is under the agranulocytes, we are having two. One is lymphocyte and another one is the monocyte. Is that clear to you? Now this WBC part is very important. Why? Because this WBC are called the police force of our body, or the soldiers of our body, or the mobile force of our body. Why? Because whenever we are having an infection, whenever we are having any shortage of resistance power, they provide us the resistance power of our body. They are the immunity power of our body. They help to fight us against the infection or any kind of diseases. So that's why they are called the police force, mobile force, or the soldiers of our body. And they are again having such division of components. So we will have a look of their shape and a little bit about their function. Clear? And then we will go forward with the platelets, the last one. So if I am dealing with the first one, that is the eosinophil. What is the structure? They are actually, WBC are actually rounded sometimes they are irregular. So they don't have any fixed shape. They are irregular, amoeboid like this. So if I try to draw here the structure of the eosinophils are mainly bilobed structure. They are having two lobes, mainly. They are having two lobes, so they are called bilobes. And if I want to draw a basophil, they are having three lobes. So nothing, I will draw three lobes over here. Second lobe and the third lobe. They are having three lobes. And if I talk about the neutrophils, they are multi-lobe. That is, they are having more than three. So here these are all type of eosinophils, basophils, neutrophils. These are all the different types of WBC. In a blood smear, I am also at the same time showing you the shape of the all types of corpuscles. Clear? So what is their function? So why they are called the granulocytes? Because they are having some granules in it. That means granules means they are having the granules like this. Cytoplasmic granules. As the neutrophil are stained by both the acid and the basic dye, so they are called neutrophil. Eosinophil and basophil. Eosinophil is acidophilic cells, and on the other basophil cell, they mainly takes a stain of the base stains. Clear? Anyway, if I come with the agranulocyte, that means they don't have any granules in the cytoplasm, and they looks like this. The monocyte is somewhere round in shape, but lymphocytes are somehow round in shape, and the monocytes are having kidney-like shape. Clear? This is about the lymphocytes and the monocytes and lymphocytes. So here I have drawn the 5 types of WBC, both the granulocytes and the agranulocytes. Sometimes maybe in a skill-based question, it can be given that you are having a figure like this, and you can be given to just level the parts, which one is what, and then the questions can come out that what is the function of A, B or C or 1, 2 or 3 level as in your question paper. Clear? So from a smear of blood you can have a question. Got it? Now what is the function of these all WBC. So eosinophil mainly they destroy the bacterial type of cell. They have the capability of destroying the germs. On the other hand, neutrophil, they are having the phagocytic activity. You know the phagocytic activity, that is engulfing the microorganism. They have the power to engulf the microorganism. On the other hand, basophils, they are mainly concerned with the binding of the antibodies. So all of them are involved in somehow the immunity power of our body. On the other hand, the monocytes and the lymphocytes, both the monocyte and the lymphocytes, this monocyte and the lymphocytes, they are mainly among with that, they are also mainly concerned with the phagocytostic and the immunity power of our body. Binding lymphocyte mainly helps in production of the antibodies in our body. On the other hand, monocyte mainly have the phagocytic activity like the neutrophils. Apart from that, here basophil is very important. Here that I will explain in platelets, but I am giving the term over here so that I can remind you when I am going to teach you the platelets. The important term over here is the heparin and histamine. And why I am writing these two important terms in the corner, I will let you know when I will deal with the platelets. Clear? So more or less I have said the granules, about the granules, the eosinophil, the basophil and the neutrophil, the granulocytes, lymphocytes and monocyte. Lymphocyte is again divided into small lymphocyte and as well as large lymphocyte. Clear? This is all about the corpuscles. Now coming to the greater part, that is the platelet. They are devoid of nucleus. They don't have any nucleus, and they are the megakaryon cells that is found in the bone marrow. Before going to that, I would like to have an important discussion over here, important term, that is called diapedesis. What is this? Now the blood is flowing. Suppose the blood is flowing within the blood vessels. Some of the irregular shaped WBC have the tendency of coming out through this blood vessels because of their amoeboid movement. They can move like amoeba, so they have the movement like amoeba. Sometimes they have a tendency to come out from their own space and comes to the interstitial space. At that time, such movement is known as diapedesis. That is the movement of the amoeboid movement of the WBC, due to which they come out of their own respective blood vessels, these WBC cells, and comes out. That is known as the diapedesis. That is also important for a short question. Now the last one that is left is the platelets, and I will go with that, the last one. I hope you have understood about RBC, WBC, and the last one, that is the platelets. So if you want, you can make a copy of this. This is a rough note for you. So if you like, you can copy it down. I am giving you 5 minutes for that. Just do it. In the meantime, I am rubbing the board for explaining you about something more regarding the platelets. Okay. Now coming to the last one, that is the platelets. Now you know that the platelets are very important in our body. Why? They are mainly concerned with blood clotting. The main function is clotting of the blood. That is, they helps in blood clotting. And here in class 10, you have to read the mechanism of blood clotting. How do they do so? How do they perform the tasks of blood clotting? So suppose one vessel has been ruptured. Okay, so I am writing over here the ruptured blood vessel. Ruptured blood vessel. The blood vessel is being ruptured. Then what will happen if the blood vessels will rupture? Immediately when the blood vessels will rupture, the platelets will come into action. Who will come into action? The platelets will come into action. For what? Why the platelets will come into action? They will come into action for releasing an enzymatically substance known as thrombokinase. What is thrombokinase, or you can say thromboplastin. Clear? This thromboplastin will take the help of some factors. One of such factors is known as the calcium ion. They will combine with the calcium ion, and then they will do the second step of work, that is they will again convert the inactive substance called prothrombin to active thrombin. Clear? So they will convert the prothrombin, it is an inactive state, the calcium and the thrombokinase that will be released in concern with the platelets. When they will see a blood vessels are ruptured, the platelets will come into play, they will release thrombokinase and thromboplastin, or then they will add up with the calcium ion, and along with this too, they will convert the prothrombin to active thrombin. Now this active thrombin will do the next step of work. What they will do? They will, in the next step, they will convert this thrombin will convert this thrombin will convert the fibrinogen to active fibrin. What they will do? They will just convert this fibrinogen, this is the soluble protein, okay, in the plasma, but they will convert it to a mesh-like structure, a mesh-like network, that is called they will convert it to a mesh-like network that is called the fibrin, in which all the blood corpuscles will be trapped. So, as a result, due to which